Everyone, I am Marie Angelica Hidlaupon from BS Ed Science 3, and today I'll be discussing the fascinating world of cell division. So, for today's video, we'll explore how cells multiply and maintain life through two identical processes, as we all know, the mitosis and the meiosis. So first, basically, we'll look at mitosis, which is a process that allows cells to grow, repair, and replace themselves, ensuring that each new cell is an identical copy of its parent. And then second, we'll uncover the unique process of meiosis, which also plays a key role in sexual reproduction by creating genetically diverse cells. And later on, we'll also be discussing why cell division is important. So, by understanding these processes, we can see how life continues, adapts, and evolves. So first, let's start by exploring mitosis. So mitosis takes place in regular body cells and it keeps cells living and growing. And also, in addition to that, mitosis is a type of cell division that results in two daughter cells. So again, it results in two daughter cells, each having the same number of chromosomes as their parent cell. So it is crucial for growth, repair, and asexual reproduction in multicellular organisms. So mitosis is consists of several stages or phases. So these are the basic phases of a cell's life. First, we have the interphase, the prophase, the metaphase, the anaphase, the telophase, and lastly, the cytokinesis. So first, let's start by the interphase. So interphase is the longest stage of a cell's life. It is the time spent between divisions. It produces all materials required for growth and preparation for cell division. So interphase, although it is not technically a part of mitosis, interphase is just a preparatory phase where the cell spends most of its time. So again, as what I've said earlier, this is the longest stage of a cell's life. So therefore, it is where the cell spends most of its time. And it is subdivided into three stages. First, part of the interphase is also known as the G1 or the gap 1 phase of the cell cycle. So, in G1 or in gap 1, it is where the cell grows in size and it also produces RNA and synthesis proteins necessary for DNA replication. So, this is the G1 or the gap 1, the green one, as you can see on the screen. So, in other words or in short words, it is where in the G1 or in the gap 1 where the cell grows in size. So, it is the growth phase of the cell. And second, we have the S phase of the cell cycle, the orange one, as you can see again on the screen. It is where the cell replicates in DNA, resulting to two identical sets of chromosomes. And also, in the S phase, it is where the replication of DNA happens. So, synthesis phase, when the DNA duplicates. And lastly is the G2. It is just a checkpoint to make sure that the DNA is correct. And also in the G2, the cell continues to grow and produces proteins required for mitosis. So and after that, after interphase, the mitosis happens. So mitosis, the last part of the cell cycle is called mitosis and has 4 phases during which the cell divides into 2 cells. So the first one is the prophase. So prophase, the cell begins the division process. So the nucleolus disappears and the nuclear membrane breaks apart. And also sa prophase, the chromatin or the material chromosomes are made of condenses into visible chromosomes, so each consisting of 2 sister chromatids. Then the nuclear membrane begins to disintegrate and the mitotic spindle starts to form from the centrosomes. So in addition to that, the chromosomes become visible, as what I've said, and also the spindle apparatus forms at the and attaches to the centromeres of the chromosomes. So the centromeres is in the middle part of the chromosomes. And I want to zoom in. This one. This is the centromere, the yellow one, as you can see at the middle. And second is the metaphase. So metaphase is the second phase of mitosis. So the nuclear membrane is completely gone. So as you can see, the nuclear membrane is not visible anymore in the metaphase, and the duplicated chromosomes line up along the cell's equator. So the chromosomes align along the metaphase plate or in the center of the cell, and the spindle fibers attach to the centromeres of each chromosome. And for that is the metaphase and the spindle apparatus. The spindle apparatus is really not that visible. And the third phase of mitosis is the anaphase. So diploid sets of daughter cell chromosomes separate. So they are pushed and pulled toward opposite poles of the cell by the spindle fibers. So in anaphase, the spindle fibers shorten. Again, so the spindle fibers, these are the spindle fibers. So as you can see, the spindle fibers shorten, pulling the sister chromatid apart to the opposite poles of the cell. So this ensures that each new cell of the daughter cell will receive an identical set of chromosomes. And then next is the telophase. It's the nuclear membrane and nucleoli reform. So cytokinesis is nearly complete. And after that, the cell plate begins to form and the cell prepares for its final division. So, and lastly is the final stage of mitosis is the cytokinesis. So the cytoplasm, organelles, and nuclear material are evenly split and into two new cells are formed. So, or in other words, the cell membrane pinches in and divides the cytoplasm into two separate daughter cells, so each with a complete nucleus and identical genetic material. So each of the daughter cell have a complete nucleus and identical genetic material. So after that, the two new cells, each exactly like the other, are called daughter cells. Because we are talking about mitosis, so it produces two identical daughter cells. So that is the process of mitosis. So now we have the two new cells, each exactly like the other. So, or they are called the daughter cells. So that's it for mitosis. So now we'll be talking about meiosis, why we are who we are. So meiosis is also a specialized type of cell division that reduces the chromosome number by half, resulting in 4 genetically diverse daughter cells. So it is different from mitosis, because if mitosis can produce 2 identical daughter cells, so here in meiosis, it results into 4 genetically diverse daughter cells. So it is essential for sexual reproduction and occurs in 2 consecutive stages, which is the meiosis 1 and the meiosis 2. So meiosis takes place in the gametes of an organism. So people have a chromosome count of 46. So when an egg joins a sperm, the count must stay at 46 to remain human. So the egg can only have 23 chromosomes and the sperm can only have 23 chromosomes. But the integrity of the organism must be... What's this? I don't know, but so how does this happen? So during meiosis, gamete or the sex cells undergo a double division, so maintaining the DNA but reducing the chromosomal count to 23. So in meiosis, it should have a count of 46 chromosomes in total. So 23 from sperm, 23 from the egg, which will result into 46, which is the fertilized cell. So this is what happens in meiosis. So again, as what I've said earlier, in meiosis, it is divided into two stages. So first, let's take a look at the meiosis 1. So what happens in meiosis 1? So just like mitosis, it is also have the prophase, the metaphase stage. So the original gamete, as you can see at this screen, so the original gamete, or these are the, as you can see, the chromosome condense and the homologous chromosome pair up in a process called synapsis, forming tetrads. So crossing over, the exchange of genetic material between homologous chromosomes occurs, increasing genetic diversity. So the nuclear membrane breaks down and the spindle apparatus forms. So what happens to the metaphase? So in metaphase stage, the homologous chromosome pairs align along the metaphase plate, and spindle fibers attached to centromere. So these are the spindle fibers. As you can see, it is attached to the centromeres. And C, which represents the anaphase. So what happens here? So in anaphase 1, the spindle fibers pull the homologous chromosomes apart to opposite poles of the cell. So unlike some mitosis, the sister chromatid remain together. So in telophase 1, as you can see, the chromosome reach the poles. So each chromosome reach the poles, and the nuclear membrane may reform briefly. So the cell undergoes cytokinesis, resulting into two haploid daughter cells with half original chromosome number. So as you can see, in cytokinesis, it is the cell undergoes na into two haploid daughter cells. So it results into two daughter haploid cells, but it doesn't end with that. So the cycle still continues in meiosis 2. So basically, so the question now is, what happens in meiosis 2? So in prophase 1, so assume that it has now two daughter cells because of cytokinesis. So the chromosomes there condense gain. So if they had decondensed, so and a new spindle apparatus forms. So the nuclear membrane breaks down if it reformed after the meiosis 1. So second, in metaphase 2, as you can see, the chromosomes, they align along the metaphase plate. So it is basically similar to metaphase in mitosis. So again, the chromosomes align along the metaphase plate. And then third one is the anaphase 2. So what happened? The spindle fiber pulled the sister chromatids apart. So now they are at the opposite places of the cell. So because the sister chromatids pull apart, so it results in two opposite poles of the cell. And in telophase 2, the chromatids reach the poles and the nuclear membranes reform around each set of chromosomes. So what happens there? Cytokinesis occurs after that, resulting into four genetically unique haploid daughter cells. So each with the half number of chromosome as the original parent cell. So even though the mitosis produces two identical daughter cells, it is really identical, but in meiosis it produces four genetically unique daughter cells or diverse cells. So it's not the same into meiosis. So now that we've done discussing the mitosis and the meiosis, let's now I'm also gonna read this. So at the end of meiosis, the individual gamete cell has divided from one cell to four. So male produce 4 viable sperm, female produce 1 viable egg, and 3 non-functioning polar bodies. So this is for addition lang. So meiosis ensures that all living organisms will maintain both genetic diversity and genetic integrity. So now that we've done discussing why cell division, the mitosis and meiosis, we've done differentiating them. Now it's time to think or to realize or to explore why is cell division important. Why is it important, especially for us humans? So why is cell division important? So first, all living things, as we all know, are made of cells. So basically, this is the cell theory, that we living things are made of one or more cells. So the principle states that all organisms, whether they are unicellular, what is unicellular? So unicellular is only composed of a one or a single cell, or multicellular, which is composed of many cells. So are made of cells. So cells are the building blocks of life. So next is number 2. The cell is the basic unit structure and function of living things. So cells are the smallest unit of life that can carry out all the processes necessary for living. So they are the fundamental structural units of all organisms that are responsible for carrying out essential life function. So each cell has specific parts, or as we call it, the organelles that perform particular functions. So for example, sa animal cell, the nucleus, as we all know, stores genetic material. So the mitochondria generate energy, and the ribosome produce proteins. Ribosomes produce proteins because it is the protein synthesizing machine. And so another example is, let's say in plants, the chloroplast carry out photosynthesis, converting light energy into chemical energy. So this principle highlights that all biological function of an organism, such as growth, metabolism, and reproduction, are carried out by cells or groups of cells working together. And for the last one, all cells come from pre-existing cells. So this principle states that new cells are created only by the division of existing cells. So it refutes the idea of spontaneous generation, or the theory that living organism could arise from non-living matter, and asserts that cells arise only from other cells through processes like mitosis or meiosis. So let's say, for example, when a cut on the skin heals, new skin cells are produced by the division of the existing skin cells. And another example, in reproduction, an organism starts as a single cell formed from the fusion of two cells, so such as the sperm and the egg in animals, and then divides to form all the cells in the organism's body. So that's all. I hope that you learned something from today's video, and stay tuned for our next video. Thank you.